building.
Okay, yeah. Okay.
And the Caesarean section is a district in Rome.
Right, okay, yeah, I've seen that. Okay, now in the large intestine, we have some slow peristalsis moving things along. We also have something called haustra filling, okay. So the haustra fill up, these large intestine has these talia coli, which is smooth muscle that's shorter than the length of the large intestine, so it makes it puckered. Those puckers are called haustra. Okay. And so as each one fills up, it then contracts and pushes things to the next one and down the line. So that's one way we move things along, besides slow peristalsis. But the major thing is these mass movements that happen 3 or 4 times a day during meals or after meals. And that pushes things along, and then you'll have that urge to defecate sometimes during or after a meal. And of course, at this point, what are we absorbing? Water and vitamins primarily, okay? Now, most of the water was already absorbed in the small intestine. But there's a little bit more water you can absorb, and there's the vitamins made by whom? E. coli, Escherichia coli. Okay. But the feces coming out then will be largely, things you couldn't digest, and about 25%
E. coli.
Okay. So we have the mucosa, we have the submucosa where there's lots of glands, we have the muscularis, and we have either the serosa or adventitious where it's located. And remember, some things are in that peritoneal cavity, some things are behind that peritoneum, and if they're behind the peritoneum, they're called retroperitoneals. And give me some examples.
Pancreas, duodenum, kidneys, ureter, bladder.
Yeah. Ovary. I mean, uterus, not ovaries.
So which, the outer layer, the Avantia, or the beginning with S, which one is where? The outer layer of the...
serosa versus adventitia?
Yes, one's in and one's out of the peritoneum. Right?
Yeah, and I always forget. So look up my notes.
So that's not going to be on the test?
Oh, it could be. You look up my notes too.
Right on.
I'm afraid to say because I got to make sure, because I'm always getting those two mixed up myself.
Gosh,
it happens to the best of us.
Should just take it out.
Not in that. Okay? I should know it. I'm always forgetting that one. There's some things that you always remember and some things you always forget.
Just locate.
That's one of those things I'm always forgetting for some reason. And I put it down there for you, but I'm always forgetting that myself. So moving on to something I do know more about.
Okay.
Why don't you hand me that one too? I'll do all these
questions about this too. Just something to look forward to.
And I think it's down below on that one too. I think it's adventitia above and serosa below. I better check it.
It was inside and outside.
Yeah, it has.
Does the duodenum have?
It has to do also with I think.
The video 15 minutes. Good.
Pelvic cavity versus the breast.
I would disagree with that. Because the duodenum, I mean, all of that's in the abdominal cavity. I mean, we don't talk about the lungs having being retroperitoneal or
No, but I'm talking about the layers of the GI tract.
It's all in abdominal cavity, right? Except the esophagus.
No, yeah. No, same. No, esophagus is
One
not.
thing. One thing.
So, kidneys, urinary system. As far as I can tell, it's all retroperitoneal. Okay? So, kidneys, with the adrenal glands on top, abdominal aorta, inferior vena cava, and male or female? Male, because we have
prostate.
Prostate. Watch out. Here's the prostate gland. What's this little triangular region called? Trigo. And what are the landmarks of it?
2 urethral
openings and the
urethra.
opening to the urethra, okay? Also, you can see back here the
seminal vesicles.
Okay. And this, for some reason, only has one. It had 2 before this semester.
Shut down.
Yeah. Okay, let's go over some of these structures here. Okay, so this kind of duplicates. Okay, the outermost part is the fibrous capsule, and there's also fat, another layer of connective tissue, and then more fat. Okay. And inside is the
Cortex.
cortex, and then we have the
Pyramids.
between them
Columns.
columns. And the tip is called the papilla. And that's, when it comes off the tip, it's done deal. The urine is not changed. Okay, before it's a filtrate. So that's called what? Right after.
Minor calyx.
Minor calyx, okay, and major,
Major.
and then
Pelvis.
renal
Renal
pelvis,
pelvis.
and then your, okay.
Ureter.
And we have two basic types of nephrons. I'm not sure about this guy. I'll look that up. This is called what kind of nephron?
Cortical.
Cortical nephron, and this is called a
Juxtamedullary.
juxtamedullary. And what's the difference?
Well, one goes deeper.
It has a long loop, nephron loop or loop of Henle. Okay. So this one, this would be what?
The Bowman's capsule.
Bowman's capsule, glomerular capsule,
Glomerulus.
glomerulus inside, and then we have the proximal convolute tubule, descending limb of the loop of Henle,
Ascending.
descending limb, distal convolute tubule,
Collecting duct.
distal, and collecting duct. Okay, here's the fill it down here. And so this is, most nephrons are what? Cortical or juxtamedullary? Cortical, 85% or so. And there's only about 15%, but why are they important?
For concentration of urine.
Yeah, so you can change how concentrated urine is with these, especially. Down here is in the medulla, this is the cortex. Down here in the medulla, very concentrated in salt, because we pump a lot of salt out.
For water.
Now, in the ascending, it talks about it's impermeable to water, but sodium can travel through, which would go against the where sodium goes, the water follows principle.
In this case, we're not just absorbing sodium, we're pumping it out.
Okay, but so the water stays in and the water,
Yeah.
water doesn't follow in
Right,
that section.
right. In this case, it doesn't.
Okay, and then
The
also
water comes out in the descending limb because and very high osmotic pressure out there. And these would have special kinds of capillaries called vasorecta.
now with the
Around here would be the peritubular capillaries.
so does a efferent arteriole either become a vasorector or a peritubular capillary, or does some of the peritubular capillaries go down into the medulla to become the vasorector?
Some,
And do you have vasorector on the cortical with appendix?
well, they always talk about them being around here, but you must have some capillaries here, I would imagine. But they don't usually show them. But they talk about, especially the vasorecta, around these long loops of them.
So on the cortical loop of Henle, you wouldn't necessarily have, but there's got to be some.
There must be something. There must be
It's not
something.
the vasorata.
Yeah,
Okay.
but these long looping ones are called vasorecta.
So do the efferent arteriole either become vasorector or peritubular capillaries, or does some go?
I think some must be both. I think some must do
some
one,
of
some
them
must do
are
others, because you still need the peritubular capillaries too.
Okay. So
So
there's
I mean, just reasoning it out, I would think you'd have to have both.
okay. So some go one way and some go.
They don't
the
even
other way.
talk about it, but but
I know,
you
it's
have
fine,
to
I've got
have. Yeah,
questions.
that's right. I was saying it'd be reasonable to have both. Okay, so coming in here is what? Afferent arteriole. How is it different from this?
F.
And larger.
Bigger.
This is larger. This is called what?
Efferent.
Afferent what?
Arteriole.
Arteriole.
Okay. What is this?
It's the distal
Distal
convoluted
convoluted.
This is convolute tubule.
Proximal.
Let's call it the ascending limb. So this is coming in, and it's going to break into all these capillaries called
glomerular capillaries.
glomerular capillaries or glomerulus. This is larger than this. What's going to happen to pressure here?
Increase.
Yeah, so that's what we call the filtration pressure. So what happens here in Bowman's capsule, this is the parietal layer, this is the visceral layer. We're going to push a lot of fluids out to make a filtrate. So that first step in making urine is the filtration step. So we get a huge blood flow to the kidney, right? Huge blood flow. 1/4 or 1/5 of the blood goes through every minute. So, but we don't want everything going into the urine, so we're going to filter things out. The first step in filtering things out is filtering through what?
Fenestrations.
The fenestrations of the glomerular capillaries. So these are fenestrated capillaries. Everybody missed that. Everybody put down peritubular capillaries. Fenestrated.
We had to study it another time.
Oh, okay.
And
Well,
plus
then you
I
go back.
copied off all of my groups, so.
Yeah, well, that's why.
True.
Fenestrated capillaries. Well, the whole, almost everybody, with a very few exceptions, put down peritubular capillaries. No, fenestrated capillaries, very special capillaries because they have those windows.
Okay, and those windows filter out your PowerPoint says the cells, so that would mean they filter out white blood cells.
Actually, it's going to filter out formed elements in general.
So platelets don't pass through it?
I wouldn't be surprised a few could, because they're small enough, but I don't, they make it sound like it's all the...
I know, because nowhere does it talk about platelet reabsorption.
No. I think they're talking about basically all the formed elements. Almost.
Okay.
Then the next layer would be the basement membrane. That's just, again, educated guess. They don't talk about the platelets, but I would assume it's everything. The basement membrane then would filter out what?
Large proteins.
Large proteins.
medium proteins.
And then, nope, not medium.
Just large.
Large proteins. What filters out the medium proteins?
The slits in the pedicle.
The slits, okay, little slits here in the pedicles, okay, filtration slits. And that's a medium-sized protein. So what gets out here in the filtrate is plasma minus most of the proteins.
Now, albumin, we learnt, was a small protein. Well, we learnt it was the smallest protein.
Of the liver proteins.
Of the plasma, blood plasma proteins. Now, does albumin get filtered out, or is that reabsorbed?
If it is in there, it would be reabsorbed. But I don't know specifically about albumin.
It makes it through the slits.
Because albumin should not show up in the urine.
Right. Proteins in general should show up.
Proteins should not show up. Okay. But amino acids are still here.
Yes.
And sugar is still here. Glucose is still here. Okay. And salts are still here. So we go next to the proximal convoluted tubule. The proximal convoluted tubule has very specialized cells here. What are they?
We've got microvilli.
They have microvilli. What shape are they? Look at them. What shape are they? Cuboidal.
Colloidal.
Cuboidal. Who's that columnar?
They're big.
Those look pretty columnar from here.
They don't look very columnar to me.
I said that.
I did.
Unless your voice has gone up, if you're sitting on your balls or something, and she said it.
I don't have to worry about that for another couple years.
Because these are microvilli, not pedicels. So these are microvilli here. And besides having lots of what are the microvilli for?
Increasing surface area.
Increasing surface area. And what else do we have a lot in there?
Mitochondria.
Mitochondria, because we're going to do some active transport, right?
That's right.
Okay, so moving on, let's see, where do we want to go here? Okay, so the proximal convoluted tubule, what are we absorbing there? What's happening in the proximal convoluted? So we've already done a filtration step. Now we're going to do a lot of reabsorption, okay? Nutrients like amino acids and glucose, for one thing. Unless you have too many, I mean, glucose has to have a carrier. So if you have diabetes, so much glucose comes through, your carriers get saturated, and so glucose goes on through to the urine. But assuming it's a normal amount of glucose, you reabsorb it all. Because you don't want to lose those nutrients, okay? Physiologically speaking. Okay. And then we reabsorb the amino acids, reabsorb some sodium, and a lot of water. In the descending limb, lots of water again moving out into the capillaries. Ascending limb, water impermeable, but here we're going to pump out a lot of sodium chloride. Distal convoluted tubule, we're going to do lots of secretion, especially. So we're going to get rid of toxic nitrogen-containing waste, things like urea, ammonia, creatinine, things like that. Also, variable control on your hormones, right? Distal convoluted tubule and collecting duct are under the influence, of what hormones? Aldosterone and antidiuretic hormone. So antidiuretic hormone causes you to reabsorb what?
Water.
What does that do to the blood volume?
Increase.
And blood pressure.
Blood pressure increase.
Okay. So that's going to go to those peritubular capillaries. Also, aldosterone, what does it cause the reabsorption of? Sodium in exchange for potassium, but water can follow it. That